Hi, I'm Phil Mitchell and I'm Director of Integrated Biology at the Charles River site, Saffron Walton. CHDI is a privately funded, non-for-profit biomedical research organisation devoted to a single disease, and that is Huntington disease. And they operate a fairly novel model where they facilitate drug discovery focused around Huntington disease, and they want to develop a drug that slows the progression of the disease and gives real clinical benefit to the patients, and they want to do it in a quicker time as possible. And they do this through a sort of unique model of bridging the gap between academia and industry, and so facilitating that translation as quickly as possible. And they want, and they've got the capabilities, and they have projects that go from target identification, validation, all the way through to ideally delivery of a clinical candidate and clinical trials. So Charles River, we support CHDI through a whole variety of initiatives, from target identification, validation, hit identification, hit to lead, all those preclinical drug discovery efforts, including ADME, DMPK, and biomarker assay development, to enable demonstration of proof of concept in relevant Huntington disease animal models that Charles River can provide, with the aim of delivering a preclinical candidate for the benefit of Huntington disease patients. Working together with CHDI, we implemented in vitro neurobiology platforms, both primary neuron and stem cell derived neuron. And then we've also enhanced our biomarker assay development and screening capabilities so that we now offer that as a service. And together, working, we've enhanced our knowledge of the disease, both at the molecular level through to how it impacts on the patient. So we have a great deal of scientists with that expertise, and we know who are the key players within the Huntington field, and we've got a good overview of the Huntington drug discovery field at the moment. So we can apply multidisciplinary teams to any client project, and it can be from the in vitro biology through HTS, hit identification, and that could be through phenotypic screening or target-based screening, and then thinking the drug discovery element of medicinal chemistry and ADME, DMPK, through to key experiments like biomarker assay development to give confidence that the compounds are going to show an effect, and we can measure an effect in a relevant Huntington disease animal model, and then deliver, ideally, preclinical candidate to that organisation.